Hello, I'm Amanda McGill Johnson with the Nebraska Coalition for Life-Saving Cures. Our mission is to promote, support, and advocate for life-saving research. And this particular program is created to really elevate and draw attention to the really awesome research happening around our state. We are fortunate today to have Dr. Iqbal Ahmad here with us from UNMC. He's a developmental neurobiologist, and he spent more than 30 years studying the inner working of the brain and its relationship to the retina to understand what leads to blindness. Thank you for joining us.
Thank you.
If anybody who's viewing this live wants to, you can put questions in the comment box and I'll be monitoring them during this interview. First, Dr. Ahmad, talk to me a little bit about the relationship between our eyes and our brain.
Well, Amanda, not many people know that the retina, which is a very delicate tissue at the back of our eyes, is an integral part of our brain. And so it develops from the same primitive neural stem cells that give rise to other brain regions. For example, other brain regions like, say, for example, hindbrain, which regulates our respiration, midbrain, which facilitates our movement, and forebrain, which regulates our social behavior and cognition. Retina is also composed of neurons and glia. And glia are the support cells that facilitates the function of neurons in the retina and in other part of the brain. And so that is basically the relationship the retina has with the brain, but in adult, it is integrally connected with the higher center of the brain for the perception of vision. So retina function like a receptor for light. It captures the light and converts the light into chemical signals. And that chemical signal is conveyed to the higher center of the brain through optic nerve, which is a part of the retina, for example. And within the higher center of the brain, the chemical signals are decoded so that we can perceive an object in our visual field. So structurally and functionally, it is highly connected with the rest of the brain. And if you try to draw an analogy of the circuit, we can think about something which is very familiar to all of us, that a TV antenna, which is connected through cable to a TV set, right? And the signal in the TV antenna is being conveyed to the cable to the TV set, where the signal is converted into image that we see. So that is a structural and a very simple example to understand the relationship between the retina and the brain. But I would like to add a little bit more to it, which not many people are familiar, that a lot that we know about how the neurons in the brain communicate with each other and process information so that we can execute myriad error-free functions every second, every hour, throughout the day, such as ascribing color to an object. Apple is red, banana is green, grass is green, and to the extent that when you throw a curveball and we don't lose our balance, has come from understanding how the retina functions. So retina is a window to our brain.
Those are great descriptions. So what do we know about what does lead to blindness?
Actually, there could be several causes of blindness depending upon where the disease is happening within the eyes. For example, if the transparency of the lens in our eyes is lost, then the light will not be able to efficiently in a proper way so that the vision will be lost. Another way the blindness can happen is through the disease of cornea. Cornea is the front end of our eyes, which are transparent. And if we lose the opacity of cornea, the transparency of the cornea, because of the disease or injury, again, the vision will be lost. Then there are blindness known as age-related macular degeneration, which a lot of us are familiar with. Old folks in our family suffer from those. And glaucoma. These are the blinding diseases due to degeneration of specific kind of neurons within the retina that leads to blindness. So there are many causes. Some of the blindness and vision loss are reversible. For example, you can replace the lens and the transparency is restored and the vision is restored. Other kind of blindness are irreversible, glaucoma being one of them,
Okay.
and it is still an intractable blinding diseases.
Of which you're trying to work on.
Trying to understand and work on, yes.
So in the last couple of years, your lab has made some advancements in the understanding of the role of some of these particular types of cells in the retina in the development of glaucoma. Talk to me about what you're learning from these cells.
Yes, I think there's a lot of learning to be done in a simple form. When we look at the retina, it is a simple model of the brain. It contains only 7 different kind of cells as opposed to highly complex other region of the brain. And all these 7 cells are working collaboratively with each other so that we can see an object properly. And so one class of the cells known as photoreceptors, as the name signifies, captures the light that is reflected from the object that we want to see and convert the light into visual signal. So you can imagine that if those cells degenerate, then blindness will occur, signals will not be generated, and we will not be able to see an object that we want to see. And that happens in age-related macular degeneration. So there are 2 different kinds of AMD, wet kind and dry kind. I will not go in detail in that. If you have a question, I can answer a little bit later on that. So once the signal is generated, it has to be transported or conveyed or relayed outside the retina to the brain. And the cell that does that function is known as a retinal ganglion cell. It's a mouthful, so we use the acronym RGC. So the RGC are the cells which relay the chemical signal for vision through optic nerve to the higher center of the brain. So if RGC degenerates, the signals are generated fine, but they're not able to be conveyed to the brain so that the vision could be perceived.
So the cable is broken. Going back to the TV antenna, the cable is broken.
The cable is broken. And so there is a disconnect between the higher center of the brain and the retina. And that's how we lose our vision. But for your audience, it's very important to convey to them is that the degeneration of RGC happens very slowly, imperceptibly. And we do not know that we are losing our vision. And till we go for ophthalmic examination, and then it is discovered that we are losing vision. So that's why this disease, the glaucoma, is known as silent robber of vision. The 3 million patients that we have of glaucoma, half of them did not know that they're losing vision. Half of them. So that is a disturbing feature of this disease. And when we talk about RGC, we don't have any idea yet why these cells degenerate to begin with. We have no idea. And we know for sure that there are some risk factors like high pressure within the eyes, the race, family history, and myopia. Those are the risk factors which are associated with glaucoma. But why the cells die is still a mystery. We do not know. So the race is to find the cause behind the degenerative changes in RGC. Our lab has a different take on it. We think that the disease is due to the developmental defect of RGC. So there is an abnormality in RGC to begin with.
Okay.
And that abnormality is unmasked as people age and when it is paired with certain risk factors. And then we find that RGCs are degenerating and we are losing vision.
So how do you check that idea or test that idea? And go, are you able to do that?
So when we pose this hypothesis that that may be the reason, those ideas could be tested if in animal model,
Okay.
right, in rats and mice, which are usually worked in the laboratories. But the moment you ask this question in context of human, you can't test that proposition because the majority of RGCs are born during the first trimester of the fetal development. So we have no access to study that phenomenon at that early stage of the disease. We can't do it. So to circumvent this process, we use a technique by which we can generate stem cells from the blood of a patient or skin or even the cells in the urine of a patient. And that technology is known as induced pluripotent stem cells technology, for which Shinya Yamanaka got the Nobel Prize, and which has revolutionized how we understand a disease, complex disease process, and formulating techniques and approaches how to actually treat them. So we are using that particular technique to test our hypothesis that the human RGC, the disease in glaucoma, actually develops in the fetal situation. So how did we go about it? So we got the blood from a patient suffering from glaucoma and also the blood from the age-matched healthy donor.
Okay.
And from those two blood samples, using the induced pluripotent stem cell technology, we generated the stem cells. Once we had the stem cell in our hand from the patient and from the healthy donor, we next converted these stem cells into RGC. And to convert it, all the information came from the study that we have been doing for the last 30 years. Everything was, those information came very handy. And at each state of generating the RGC in a dish, we asked the question, do we see any difference between the patient-specific RGC and the healthy donor RGC at different level? Do they look different? Do they function different? Do they have different gene expression? And we were surprised to find out that every stage of development, the patient-specific RGC looked abnormal comparison to the healthy RGC. And that was very satisfying, but at the same time, it told us that it may be possible, as we posed our premise earlier, that the disease actually occurs fairly early. And it is the constellation of risk factor. When it pairs with this kind of abnormality, then the degenerative changes happens. So the model that we created for glaucoma using stem cell technology in our lab is allowing us to understand what is the molecular basis of the disease. And if we understand that, then we will be able to diagnose this disease sooner and may formulate some hypothesis by which we can our approaches to treat this disease in a clinical setup. The other question that you just we talked about the analogy between the TV antenna and the TV set, the cable, it's the optic nerve. So using our model system, we are asking the question that if optic nerve degenerates, can we regenerate it, right? And using this model system, we have a fairly good evidence that we can do it. So the study that has been done in animal model, many of the study that is done in rat and mice do not pan out in clinical setup. So the animal model sometimes is not very well go. So we have to confirm all those study in human context. And it is panning out very nicely using the model system that we have created in our lab.
Do you know if having this abnormality in these cells necessarily means you'll get glaucoma or blindness, or just more likely given other circumstances?
So that's a very good question. And so we are hypothesizing, our premise is the abnormality is there to begin with, right?
Mm-hmm.
And in our modeling in a dish, that turned out to be true in case of one patient. So we have to reproduce this study in many different patients. And I must tell to your audience that glaucoma is not one disease. It's a constellation of diseases. And what is common in the constellation of diseases is that the RGC degenerate no matter what. That's the commonality. And that's what we are looking at. At this point of time, what we have is various risk factors. So if you have increased pressure in your eyes, which is associated with the most prominent case of glaucoma known as primary open angle glaucoma, that's where you find increased intraocular pressure. But the conundrum is that there are 10% of glaucoma patients who do not have increased eye pressure. Still, their RGC degenerates, right? Mm-hmm. So if we consider all of these, we are still scratching the surface to begin with. But we hope the model system that we have created will incrementally add to our understanding why these cells, in particular, die in this particular disease, a constellation of diseases.
Mm-hmm. Yeah. So do you think that your research in regenerative eye healing will have applications beyond or to other systems in our bodies?
Well, I think yes. The reason I'm saying is that because the results that are coming from studying different brain regions, for example, Parkinson's disease, stroke, ischemia, and all that, the information that is coming from those studies are very helpful for us in understanding what's going on and coming up with approaches to treat those changes. And I think it's vice versa too. The information that we are generating within the retina is directly or indirectly applicable to what's happening in another part of the brain in terms of disease process, in terms of normal function or abnormal function. So it's a two-way street. We'll be benefiting no matter what. But if you look at it from a general perspective, brain and the retina, and other part of the body too, there are 4 different approaches that we can have in fighting the degenerative disease. So first approach is very simple, is that if you know a particular group of cells or neurons within the other brain region or within the retina, they're dying, you try to protect them from dying using chemicals, drugs, or any pharmaceutical. That is called neuroprotection. And that has been tried within the retina for blinding diseases in animal model. And in animal model, neuroprotection has been remarkably successful. But when they tried the clinical application, the success was not to the extent that they saw in the animal model, right? The other approach could be that if we know a gene, mutation in which leads to a disease process, then a simple approach will be to, I'm calling simple, it's not that simple. The approach is complicated, to correct the mutation and then rescue the cells from dying. So I will just communicate to your audience that fairly recently, a particular kind of blindness called Leber congenital amaurosis has been treated or tried to be treated using CRISPR-Cas9 gene editing technique. And it has been successful, so that we hear. So these approaches are going to be much more available in terms of efficacy and safety, so that the cells could be rescued from degenerative change. The third approach, everybody knows about it, transplantation. Why don't you transplant healthy cells and rescue and replace the dead cells? That's also being tried out. But the one that excites me and excites the scientists elsewhere in the world is to tap into the body's inherent capacity to heal itself by regeneration, right? If we can do that, we don't have to do any transplantation. We don't have to do any neuroinjections. And transplantation has immune rejection and new plastic formation and all those kind of complications. So why don't we tap into our own body resources? And most of people know that the part of the body which goes through regular wear and tear is regenerated continuously. Your skin, my hair, cornea, and even the internal organs which are exposed to outside, for example, intestine, olfactory epithelium, which allows us to smell, is continuously being regenerated on a continuous basis. Regeneration also happens within the brain. We come to know in the last 20 years. But those regions are very well circumscribed in two parts of the brain. One that lines the cavity of the brain, the ventricle, and the other part of the brain that functions for formation of memory. Those are the two regions. What other labs and our lab has demonstrated that the glia, the support cells in different part of the brain actually have a dormant stem cell property. So if we can coax those dormant stem cells to generate neuron within the brain, don't you think that will be a much better way to go about it? So what we demonstrated a couple of years ago, that in an animal model of age-related macular degeneration, that we can recruit the support cells within the retina, called the macular glia, and cause them to become photoreceptors, the cell that die in AMD. It was very exciting, but the problem was, indeed, they convert into neuron, but they convert very, very inefficiently. That is not good enough for clinical approach. So the race around the world in different labs is, how can we coax the support cell, the glia, to make neuron efficiently, reproducibly, so that a clinical approach could be done. So that's the one on which I am putting a lot of my hope on. And because that's very exciting too.
What is like a normal day of work like in your lab? How do you go about doing this research?
So if I describe how the normal routine is like, which changes depending upon what stage and where we are, whether we are successful or not in our experimental approach.
Interested in pursuing something like this, what would it be like?
So I have 3 young postdocs or young scientists in my lab who are working at different projects which are overlapping. So the day begins by looking at the cells which are being cultured in a dish, that how they are behaving, and feeding them at different stages of experiment. Once that is done, then we group together, discussing experiments, expected, unexpected results, how they're coming about. And many, many times it happens that there are many problems, things are not going well, so how we troubleshoot it. That is an exciting part and challenging part of lab routine. And if that is the day for transplantation, a lot of time goes in preparing cell for transplantation, then do transplantation, which take a couple of hours, and so on. In between, we try to find time to read literature, to keep abreast of what's going on. So sometimes it is boring, but most of the time it is exciting. and more so when you get some results which uh, uh, are good or some are complex to make sense of, you know.
What got you interested in becoming a scientist and in this field?
You know, uh, I'm intrigued by brain, like many other people. I'm very intrigued by brain, that uh, how it is put together and how it functions. It is amazingly, amazingly complex, and it is a mystery like no others. So I say that for a person, for each person, the brain is the center of the universe, actually. The reason I say that, because give you an example. If I'm going to have a stroke which affects the right parietal lobe of my brain, I am going to lose the world on the left side of my visual field completely, completely. My retina is fine, my sense is fine, the world is gone, it's not there. And if I'm eating from a plate of food, I'm only going to eat from half the side of the plate of the food and not the other side. It doesn't exist in my mind because of that. If I have some kind of problem in the ventral part of my brain, which actually recognizes the object, I will lose the color from my world completely. The entire world will become gray to me. As opposed to if I have a problem on the dorsal side of the visual circuitry, I will not be able to guess how fast and how slow the car is coming at me. If I'm pouring a cup of tea, I will keep spilling the tea because I don't know whether it is full or not. There are many, many examples like that that intrigues you, that the world is within your brain. So that's what actually interested me in the first place, to come to research and be and work at the bench.
That's really cool. What advice, as we approach about 30 minutes here, what advice do you have for young people who may be interested in a career like yours?
Well, you know, the commitment to science begins with curiosity. So I will tell young people, bring your insatiable curiosity, but also bring your common sense and strong work ethics. It's very important. And and also they should heed, all of these young scientists should heed the dictum of Albert Einstein, who he said that many people, I'm paraphrasing it, many people think that intellect makes great scientist. It is the character, you know. So I think that actually is very important for young scientists to think about.
What a great note to leave on. Thank you, Dr. Ahmad. Do you have any other parting thoughts you'd like to share?
No, I don't. If you have any question, I will be more than happy to answer those.
Yeah, we'll make sure to attach follow-up information if folks do have additional questions that they'd like to send your way, they can reach out to us. And
Wonderful.
with that, we'll wrap up our session. Everybody have a great day.
You too. Thank you very much.